So the final exam will include, it's comprehensive, so it's going to include everything before, but it's going to focus on the urinary, endocrine, and reproductive system. So think of it as two exams. One exam covering all the old stuff and one exam that you normally take on the new stuff, okay? So for the lecture, it's going to have 100 questions. Of those 100 questions, 50 of them are on the old materials. And that's the questions that you're going to see first, is the old materials. So essentially everything from exam 1 to exam 3. That's a lot of chapters that I have to divide into 50 questions. So that comes to about 2 to 3 questions per old chapter. And these are all going to be multiple choice, true, false. So no fill in the blanks for the old materials. Okay, so let's say, for example, if you really struggled on nervous system or muscles, there's only going to be about 2 to 3 questions on each chapter. Okay. Then the other 50 questions that you're going to see after you finish the first 50 is going to be on the new materials. So that's going to include the urinary, the endocrine, and the reproductive. So the reproductive is male and female. Okay, so technically 4 chapters into 50 questions. So each chapter will get about 12 to 13 questions. So these are going to be a lot more detailed. So if I were a student, I would kind of focus on the urinary, endocrine, reproductive as I review the old materials. That's just how I would approach this if I were you. But again, do whatever you're comfortable, do whatever you know works for you. Okay. So of the 50 questions for the new materials, 40 of them are multiple choice, true, false. And the last 10 questions are fill in the blanks. You will have 100 minutes to do this, unless you qualified for more. If you qualify for more, make sure you double check the settings that you do have that extra time. If not, send me an email. But on average, students will have 100 minutes to complete this. Now, yeah, we'll go with that. And then I'll come back and I'll tell you a little bit more. Then for the lab will have 50 questions. And they're similar to all your other lab exams. They're going to be, you have to identify. So 25 of these questions are going to be on the old materials. And the old materials start from the first lab, right, which is not necessarily identification, but knowing more of those terminologies. So don't forget to study the terminologies, the cell, the histology, so on. So everything from the first to to whatever we covered on lab exam 3. Okay, but a lot of students forget that terminology is on there and then they forget to study that. So don't forget to do the terminology. Okay, so 25 is on the old materials and 25 will be on the new ones. So the urinary and the male and female reproductive. And you will have 75 minutes to do this one. Okay. So for both of these, so both for the lab and the lecture, it is really important that you do have the webcam on. And actually, you can't even take it without the webcam. So webcam and the lockdown browser is required. Okay. Make sure you do, and it's more important for this one than any because I actually have to watch all of them, the environmental scans. So the other ones, I only watched them, your other exams, I watched them if respondents flagged you. But for this one, the environmental scans, I have to watch all of them. So make sure you're doing an appropriate environmental scan. Does anyone have any questions on the logistics or the requirements for these two last sets of exams? So same thing for the lab, spelling counts. And make sure to, if there's a pair of something, to include left or right. Make sure you're writing out the full name. Like proximal to phalanx. You know, you have to have all the parts of the names in there. Any questions? Okay. So there is a little difference from the regular routine of how when you take the exam 1, 2, and 3. So for your final exams, they're going to be available from Friday to Sunday. Okay. So you don't have until Monday. And that's not a choice I could make. And the reason for that is because Sunday is the last day of class. So that's the last day you can input into Canvas. Okay. So make sure you take your final exams the latest by Sunday. Okay. So you'll have until 11 p.m. Central Standard Time on Sunday, not Monday. Okay. So you have only until Sunday to take the exams. It will open from Friday though. So from Friday to Sunday, you'll be able to take the exam. No questions? Any questions? Okay, show a thumbs up if everything makes sense. Thank you. Because sometimes I'm not sure if my audio is going through. So it's always good to have some feedback. Okay, so if you do find that you have questions, please ask. Okay, all right. So I have 2 review sessions, and this first one I'm going to focus on the urinary and endocrine, and then on the last one because Savannah had asked about reproductive. It is a complicated one, so I want to make sure I save a whole hour for that one. So I'll do the reproductive in the evening session. So for this morning session, I'm just going to do urinary and endo, and then evening session I'll do repro, and both of them will be recorded. And I will try to get them up by tonight, latest by tomorrow. Okay. All right. So we'll get started. So urinary is also a complicated one. Some of the things you need to know in terms of how to prepare, you need to be able to list and describe the functions of the kidney. It includes filtering the blood. And oftentimes we think filtering the blood is just that it cleans the blood. Okay, but not only does it have to clean the blood, but you also have to understand how it removes the waste. Okay, I use the analogy of like a clothes washer. Okay, so you put your dirty clothes in there. It spins it and it cleans your clothes, but it also has to separate the dirt, right? So not only is it cleaning the clothes, but it's separating the dirt. And what remains then is the clean clothes. So you need to take the clean clothes out, but you need to also have a process to take the separated dirt out. Okay, and that's what your kidney does. So when it filters the blood, it's cleaning the blood, but it also has to be concerned with how it's going to remove the waste. And through the process of filtering blood, it also regulates water volume as well as pH. Okay, and that makes sense, right? Because a major component of blood is water. So it's going to determine how much water you remove and how much water you retain. And because it controls the water and the substances that are removed, it also regulates pH, because remember, pH is a measurement of hydrogen ions. So if your kidneys remove the hydrogen ions, then you're going to change the pH. If it increases how much hydrogen ions you keep, then it's going to influence the pH that way. Okay. So these are the main functions of the urinary system. You'll need to be able to tell me the gross anatomy of a kidney. In other words, what can you see with your eye? Okay. You'll be able to see the fibrous capsule. The hilum is not an actual structure. It's more of a space. Okay, and that's the space in the kidney where things can enter and exit. You also find the associated blood vessels, the renal cortex, the renal medulla, the renal pelvis. Now, there are structures that you cannot see with the eye that is also a part of the anatomy of a kidney, and that's the nephron. Okay, so for the nephron, make sure you understand what the renal corpuscle is. And technically, the only part of the renal corpuscle that's a part of the nephron is the glomerular capsule. Okay, and that's going to continue and become the renal tubules. The renal tubules has three distinct parts: PCT, the proximal convoluted tubule, the nephron loop, and the DCT, or the distal convoluted tubule. Now, some books say that the collecting duct is a part of the nephron. Other books say it is not. Your book says it is not a part of the nephron, but I added it in there just to kind of show you that it's the pathway that leads out. Okay, so for each of these structures, not only should you know the structure and its function, but you should also know the lining. Okay, now again, the lining can be difficult, but don't. So it's easier to understand how the tissue lining connects with the function and the structure, rather than memorizing, okay, this one has this tissue, this one has this tissue, this one has this type of tissue. It should make sense if you're connecting tissue lining to structure and function. Okay. And then you also want to know about the micturition process, which is the process of urine formation. Sorry, urine formation is separate. The micturition is down here, is of how you actually eliminate the urine. So you want to make sure you know the process of urine formation. What are the main steps in urine formation? So what are the main steps in urine formation? You have to start out with what first? Think of the washer. What does it have to do? Remove toxins for sure. Okay. So we'll say that removing of toxins is part of the filtration process. So that's the first one, filtration. What's the second one? Not necessarily a second one, but just what's another function of urine, or not function, but what's another step in the process of urine formation? Reabsorption, excellent. Yes, perfect. So reabsorption. Now look at the term reabsorption. It's called reabsorption, absorbing meaning you're taking it into your body, right? So the term absorption, you've already learned about that. Where or what body system did you learn about absorption the first time that you actually took substances into your body? Yeah, perfect. Exactly. So we talked about absorption in the digestive system. Okay. So we talked about how it was absorbed from the digestive tract in through your bloodstream and the lacteals into your actual body, right? So that's the first time you have absorbed it. So now the substance is already in your body. You moved it to the urinary system and now you're taking it back in. And that's why it's called reabsorption, because it's technically the second time you're bringing it back into your body. Okay. So that's why it's called reabsorption instead of just plain absorption. Then another process of the urine formation is, you're absolutely right, secretion. So let's say that you filtered all this substance into your kidneys, right? And you realize that, oh, during that process of filtering, you just drank like 5 gallons of water. Is your body going to be full of that 5 gallon until it goes throughout your whole body again until it gets to the filtration state again? No. You have a third process called secretion, where it doesn't necessarily have to go through filtration to be able to move into your kidney. Okay, into the nephron to be excreted out of the body. So that's what secretion is. You can think of secretion as kind of a last minute attempt to get rid of things that you have too much in the body, but you don't want to have to go through the entire body and back to the filtration process. Okay. All right. So filtration, reabsorption, secretion. Now your book does list another one. What's another one? These are the three main ones, though. But there is a fourth one that your book has decided to kind of pull out of reabsorption and secretion. It does have to do with blood volume, definitely. But what do we call that? Yes, thank you, Savannah. Water conservation. Perfect. Okay, so we're going to spend some time talking about these processes, because usually within this review session, students have the most question about that. Okay. Then you also want to make sure you're studying what the ureter is, what's the urinary bladder, like the trigon, what makes the trigon, the urethra, what lines the urethra, what's the function of the urethra. So make sure you know the structure of these parts, the function, as well as the layers. And then lastly, the micturition reflex, or the micturition reflex. Okay. So in the micturition reflex, this one, I I think a few of your class members did a video on that. So make sure you take some time to look at your peer videos, because they were pretty good. Okay. All right. So that is it for the urinary system. Questions before I move on? Okay. So we'll start with the gross anatomy. So what's number one? Perfect. The adrenal glands. Exactly. And if this was on your lab, you would need to indicate whether it's right or left, and it is the left. How about number two? Perfect. Number 3. Perfect. So these are the ureter. Number 4. Ah, so you gotta be careful, okay? And if you did this on the lab quiz, you would have known that you got deducted half a point for this. So technically, it is the urinary bladder. If you just use the term bladder, you will only have half a point, and the reason for that's because we have several bladders, right? We have the urinary bladder, we have the gallbladder, and we have other sacs that contain things in the body. And that's why you have to make sure you use the term urinary bladder, okay? How about number 5? Urethra, very good. Okay, all right. So you can see that if this is the kidney right here, okay, and we pull out this portion right here, you can see that it looks kind of like this. We stretched it out. And so on, okay? So this is now, we left the gross anatomy, in other words, things you can see with your eye, the things you can't see with your eye. So what we're looking at now is the nephron. And nephrons are functional unit of the kidney. So when we say they do all that stuff, filtering, reabsorption, secretion, it really is happening along the nephron. So the nephron is where everything happens along the kidney. Okay, questions? Oh, we should talk about the gross anatomy of a kidney too. Hold on, let me draw that out for you. Let's go through the gross anatomy of a kidney first before we go to the nephron. Okay, so I'm going to number it. So number 1 is right here. Can someone let me know what number 1 is? Very good, fibrous capsule. Then if I peel that fibrous capsule away, it's going to reveal this section right here. I'm going to color it in blue. Okay, now parts of it kind of get wedged in between. Okay, so what would this be right here, number 2? What would this layer be? Not the renal cortex. I'm sorry. Yes, sorry. The renal cortex. Yep. And then this layer right here. There we go. What layer is that? The renal medulla. Mm-hmm. Yep. Now the renal medulla does have certain parts in it, so I'm going to put renal medulla. So the renal medulla is composed of these two parts. So do you see those triangle parts that I drew in red? What are those triangle parts in red? The renal pyramids, very good. And then the space in between that it's technically a part of the renal cortex. Very good. Okay, great. Now at the end of each renal pyramid, let me zoom in. Okay, so you can see at the end of each renal pyramid, you'll find this area, which I will shade in pink. Oh, that's a little bit too wide. Hold on. Right here, right here, right here, 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 and here. What did I shade in pink? The minor calyx. Very good. So at the end of each renal pyramid, you will find a minor calyx. Now, when two or more minor calyx join together, let's do purple. Right here, 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 and here. Then what do we have? We have the major calyx. Yep. So the major calyx consists of two or more minor calyx. And then when they all come together, let's choose green. No, I already used green, I think. Let's choose gray. This whole area right here, what would that be? That's the renal pelvis. Very good. Now the renal pelvis is going to exit. What color should I use next? Do orange. Do here. And that's when it becomes the what? The ureter. Very good. Okay. So hopefully you the color helps you kind of see the distinction between the structures, the layers, and everything else. Okay. So this right here, this opening, that's the renal hilum. So it's not an actual structure, but it's where things can enter and exit the kidney. Okay. All right. So inside the kidney, you will find the nephron. It looks like this. Okay. So it's in between the renal cortex and the renal medulla. And that's what we're going to talk about next. So we'll start with the nephron first. So the nephron has the following parts. We'll start out with this complete structure. I will use, no, because the thing is already in yellow. Let's do something that will stand out, blue. Okay. So this whole structure right here is called the renal corpuscle. The renal corpuscle is made up of two parts, the glomerulus, which is the blood vessels, and not a part of the nephron, but it also includes the glomerular capsule, which some of you call the Bowman's capsule, and that is the first part of the nephron. Okay, so the glomerular capsule is, you can think of it like a baseball mitt or a mitten. It looks like this. Okay, so it has two layers. It has this outer layer and this inner layer. Okay, or the parietal and the visceral. Okay, and then in between, that's where you'll see kind of a network or a ball of capillaries. So the blood is coming from the capillaries and it's going into the glomerular capsule. Okay. So oftentimes we have what's called the filtration membrane. Some of you remember we did the respiratory membrane, remember? So this is the filtration membrane. So the filtration membrane includes these three parts. You have the endothelium of the capillary. Now, the endothelium of the glomerulus or the capillaries is unique in that it's referred to as the fenestrated endothelium. Fenestrated endothelium. And that's from the glomerulus. Okay. Now, and a capillary is thin, right? Because it only has that endothelium. But this capillary that belongs to the glomerulus is unique in that it has these little holes. The word fenestrate in German means window. Or fenestra is like a window. So it has these little holes along the capillary of the glomerulus. And that's what assists in the movement of the plasma from the glomerulus into the glomerular capsule. Now, in between them, you'll find a membrane, a basement membrane that separates them. And then you will find the lining of the glomerular capsule. Okay, so who we call, what's the lining of the glomerular capsule called? I'll give you a clue. Let me go on to the next image. This one right here. So here you can see the filtration membrane. So this one right here is the endothelium that I just finished talking about. You can see that it has these little holes right there. That's the fenestrated, right? Now, the part of the glomerular capsule that surrounds the endothelium, okay, looks like a feet. Do you see how you have to use your imagination? But here's kind of the feet and here are the toes. So it looks kind of like this. Okay. So this is a epithelium and it contains cells called podocytes because it looks like a feet, or a foot. Okay. And that's what makes up the visceral layer of that epithelium. Okay. And that's what forms the filtration membrane. So you have the fenestrate endothelium of the capillary. Then you have the basement membrane right here, the blue, and then you have the podocytes of the glomerular capsule. Okay, so let's go back to this image right here. Okay, so that's what makes up the filtration membrane: the fenestrated endothelium, the basement membrane, and the podocytes of the glomerular capsule. Okay. All right, so the glomerular capsule is the first part of the nephron. And you can see that the glomerular capsule is like a mitt that has two lining. There's the visceral lining and then the parietal lining. And the visceral lining is made up of the podocytes. Okay. Now, if we take a closer look, the podocytes, they have these extensions to them that they wrap around and they form, think of like interlocking fingers. They form what's called filtration slits. Okay, so they form filtration slits. And that's what allows for the fluid to move from the blood vessel. So the fluid, the plasma from the blood vessel moves through the fenestras of the endothelium, diffuse through the basement membrane, and then move through the filtration slits. So you can see that all together, it tries to do the best that it can to only allow certain things to move through and not all things to move through. Okay, so when we think about that, you have in blood, remember from the chapter on blood? Can someone tell me what's in blood? What would you normally find in blood? There you go. White blood cells, formed elements, perfect. Formed elements would be things like white blood cells, red blood cells, platelets. And then you have the non-formed elements, which would be plasma. So this structure, the renal corpuscle, does a really good job at making sure that the only things that should be filtered are things that can move through the fenestras of the endothelium and the filtration slits. So really, it's preventing anything large from moving through. Okay, so of these components, which one do you think then would be filtered? The formed elements or the non-formed elements? Non-formed elements, excellent. And that's why, is it normal to find blood in urine? No, you should not see red blood cells in urine. Okay, so if you do see red blood cells in urine, that means something happened, that either this membrane becomes inflamed or infected that allows large things like red blood cells to be able to move through. Okay, you should not find white blood cells in urine. You should not find red blood cells in urine, and you should not find platelets in urine. As a matter of fact, you shouldn't even find large proteins. Okay, and usually that's a sign of damage to your kidneys. What you should find in urine are things that you would normally find in plasma: water, sometimes glucose if you're not regulating your body correctly and you're diabetic. You'd be able to find drugs, hormones, waste products, because those are all things that would normally be a component of plasma. Excuse me. Questions? Okay, so hopefully that helps you to understand the first part of the nephron, which is the glomerular capsule. Okay, now the remaining portions of the nephron are all referred to as the renal tubules. Now, the renal tubules consist of three parts. This first part right here, the second part right here, and this third part right here. Excuse me. Okay. So I can get a sip of water. Can someone unmute and tell me, what do you know about number 2? What is it called? What does it do? Just any one of you. Tell us what number 2 is. Or you can put it into the chat. Perfect. Proximal convoluted tubule. Keep going. What do you know about it? And this could be open for anybody. What is it lined with? What does it do? Okay. You may not have had a chance to review yet. So, oh, yay. Thank you. Okay. So number 2 is the proximal convoluted tubule. Like your classmate said, proximal convoluted tubule. So clearly it's a tubule. It's proximal because it's the one closest to the glomerular capsule. I promise I will. Okay. So it's the one closest to the glomerular capsule, and it's referred to convoluted because convolute means it's not straight. Do you see how curvy this part is? And that's why it's called the way it is. Again, you shouldn't have to memorize it, right? Because you're like, oh, closest, curvy, and it's a tube, proximal convoluted tubule. Okay. All right. So the proximal convoluted tubule connects with the glomerular capsule. It tends to be located in the renal cortex. It is lined with simple cuboidal. Oh, I didn't give myself enough room. Darn. Okay, let me get rid of this 3. Simple cuboidal epithelium. Because you can see it's kind of thick, right? You can see that that set of tube is thicker than the one down here, for example. Excuse me. And it comes with what's called a brush border. It's got those it's got those absorptive cells. With microvilli. Do you remember that? Absorptive cells with microvilli. Where have you heard of this before? Excuse me, sorry. Yeah, excellent. Exactly. It's in your small intestine. Because what happened in your small intestine? It allows for absorption, right? So if this has the same structures, what do you think it allows for mostly? Absorption. Exactly. And that's why we say that the majority of reabsorption occurs here in the proximal convoluted tubule because it has the absorptive cells with the microvilli. So when you take the plasma and you filter it, and filtration happens at the at the renal corpuscle between the glomerulus and the glomerular capsule. So by the time you get to the proximal convoluted tubule, you're going to want to start to take back the things that you need in your body. Okay, so that's why the proximal convoluted tubule is lined with the simple cuboidal epithelium with the absorptive cells and the microvilli because you want to take back the things that you filtered in the first step. Okay, because you filter most of your plasma. That's a lot of your body water. and substance. Okay, so if you excreted, in other words, if you peed out everything you filtered, you would be going to the bathroom about you would be losing about 150 to 180 liters per day. That's a lot. Okay, that's like using on the toilet 24/7. That can't happen because we'll die of dehydration. So absorption, or more specifically, reabsorption, has to happen. You have to take that stuff, the good stuff, the stuff that your body needs, back, because you don't want to excrete it all the way out through your urinary system. Okay. So the majority of reabsorption occurs in the BCT. Why? Because it's structurally made for that. It's got the absorptive cells with microvilli. Make sense? Questions? Okay. Sorry about not my asthma, but my allergies is acting up. Things are starting to grow in Minnesota. All right. So then the next part is number 3 right here. What's number 3? The loop of Henle. I can tell you went to school a long time ago because that's how I learned it too, the loop of Henle. But now we call it, Savannah has it right, the nephron loop. The nephron loop. The nephron loop has 2 parts, A and B. Okay, so 3A specifically is going to be called the what? Yes, thank you, Anna. The descending limb. And make sure you put limb, okay? Not limp, it's limb. Let me make sure you don't make a mistake. Okay. So the descending limb, and then B is the ascending limb. And I keep on putting limb. Okay. So descending limb and ascending limb, A and B. Okay. You can see they don't look exactly the same. Okay. So both the ascending limb and the descending limb has segments where it's thick, like here, and thin, like here. So both the descending limb and ascending limb has thick regions, thick segments, and thin segments. So the thick segment, what would it make sense to be lined with? Simple cuboidal or simple squamous? The thick segments. Would that be exactly perfect? So you see, it's not about memorizing, it should just make sense, right? So the thick segments of the descending and ascending limb, you will find simple, cuboidal epithelium. Now the difference is between the nephron loop and the proximal convolvulus is that although it is simple cuboidal, it has little microvilli. So less reabsorption is happening here. Okay, and less materials can be reabsorbed here. Okay, because it's structurally not made for that. So what you'll find though is that you will find that mostly salts are reabsorbed here. Okay. Then you will find the thin segments. And the thin segments, what would it make sense to be lined with? Perfect. Simple squamous. Now here, there's not even going to be microvilli. Okay. So at this point where you'll find the simple squamous, the only thing that's really reabsorbed here is water. Okay. All right. Then now we haven't even gotten to the details of your information yet. Okay. We're just going through parts of the nephron. I'm just like adding layers of information as we move along. Okay. Then the last part here, technically it's not the last, but we'll put it has number 4. Okay. The distal convoluted tubule. Okay. What do you think it's lined with? Look at the shape. What do you think it's lined with? Yes, perfect. It is cuboidal because it's thicker, right? But here there is no microvilli. So by the time it gets to the distal convolute tubule, there's very little reabsorption happening. No microvilli. Okay. And then this 5th part right here, which is technically is not, according to your book, not part of the nephron, but it is the collecting duct. Okay. So same thing, simple cuboidal, no microvilli. All right, so those are the parts of the nephron. Okay. All right, so I spent a little bit more time on this than I had wished for. So I'm going to finish off this part by looking, so what would you prefer? Do you want me to talk about the blood vessels to kind of complete the story, or do you want me to start on your information? But I won't have time to finish it though. What's your preference? Do you want me to finish this picture, or do you want me to start your information, the first part? Okay, your information it is. So make sure then what you do is that you'll want to add in the blood vessels. Okay. I do have videos of this that I put into the supplemental videos. Make sure you watch that. So it's my class videos that I recorded because of COVID. So I do cover this in those videos. So make sure you watch those videos. Okay. But what I would have talked about next is I would have transpositioned over this what the blood vessels look like. Okay. But it seems like the request of the class is to talk about your information, and that's what I'm going to do next in this one. Okay. So in your information, you can see that you have stepwise glomerular filtration, and then you have tubular reabsorption, tubular secretion, and reabsorption of water conservation. Okay, so you notice I numbered it 1, 2, 2, and 2. 1 happens first. The glomerular filtration happens. But as you're moving along the renal tubules, you could have tubular reabsorption. You could also have tubular secretion, and you can also have water conservation. Okay, there's a beautiful image in your textbook that's figure 23, 22 right here. That does a really nice job at taking a look at that. So if you're looking for what exactly is being filtered, secreted, reabsorbed, it's right here. Okay. So everything in plasma is filtered at the renal corpuscle, which is right here. Then at the PCT, this is what's being reabsorbed. In other words, bringing back into the body. And this is what's being secreted. In other words, kind of a last minute, I didn't get it in time, so now I'm going to throw it in, through another method, which we call secretion. So you can see that a ton of stuff gets reabsorbed. Where? At the PCT. Why? Structurally, because it's got the simple cuboidal with microvilli. Then at the nephron loop, you've got water being reabsorbed, urea being secreted. You got some more reabsorption of salts right here. Okay, so that's all along the nephron loop or the loop of Henle. Why? Because structurally, you've got the simple squamous and the simple cuboidal with very little microvilli. Then when you get to the distal convolute tubule, very little salts are being reabsorbed and some salts and waste products are being secreted last minute. Okay, and you can actually continue to reabsorb water all the way down to the collecting duct. And then when it exits the collecting duct, that's when it officially becomes urine. Okay. So let's move back to the urine formation. Okay. So you can see that it starts off with glomerular filtration. Remind me, where does glomerular filtration occur? Between what and what? Glomerular filtration occurs between what two areas? Perfect. It's going to occur at the renal corpuscle, so between the glomerulus and glomerular capsule. So right here. Here's the glomerulus. Here's the glomerulus. And here's the glomerular capsule. Okay. All right. So that's filtration. Okay. Filtration travels across the filtration membrane, and you should know what the filtration membrane is. Okay. Now, when fluids are in the glomerulus, what would you call it? When fluids are in the glomerulus, what would you call that fluid? Not yet. Nope. You call it plasma. So when it's in the glomerulus, you call it plasma. When it has moved into the glomerular capsule, now you call it filtrate. So this is a test question. Okay, so you want to make sure you know what's the fluid called, where, based on where it's located. Okay, so if anytime the fluid is in the blood vessel, it's called plasma. Anytime it's in the nephron, it's called filtrate. Anytime it's in the collecting duct and onward, it's called urine. Okay, so in glomerular filtration, the substances move from the glomerulus, where it was plasma, into the glomerular capsule, where it becomes filtrate. Then that filtrate continues onward to the next part, which is the proximal convolute tubule. Okay. That fluid, if it's reabsorbed, so if it's reabsorbed, like right here, okay, if it's reabsorbed in tubular reabsorption right here, okay, it's going to go between the renal tubules and anywhere along the renal tubules. This can happen. It's going to move back into the blood vessels. And what are these blood vessels called again? Remind me. the blood vessels that wrap around the renal tubules, they are capillaries, but what are their specific names called? They're not arterioles, actually. Arterioles are larger version. Not there you go, the peritubular capillaries. Because look at the name, peritubular. Peri meaning around, tubular because they wrap around the renal tubules, right? So the peritubular capillaries. So in tubular reabsorption, the fluid is moving from the renal tubules, whether it's the PCT, nephron loop, or DCT, back into the peritubular capillaries, in which case it's going to go from filtrate and become what again? Plasma, perfect. And that's tubular reabsorption. So tubular reabsorption occurs anywhere along the renal tubules, but it's the movement that we care about. It's moving the fluids from the nephron, specifically the renal tubules, into the peritubular capillaries. So it's the fluid was once called filtrate, but once it has moved, it is now called plasma. Okay? But let's say along the way, like I'm doing my laundry, I put everything into the washer, but I found one sock. I'm not going to stop the washer and make it go back all the way to the first step. I'm just going to throw that sock in, wherever the step is. So that's secretion. Secretion is like last minute, you're like, oh no, I have some extra stuff I need to get processed. And I throw it into the renal tubules without having to go all the way back through the renal corpuscle. Yeah, I love that analogy too, because that actually happens to me all the time. I find like loose clothing that I just like whoop, whoop, right back in, regardless of what process of the washer it's in. Okay. So the next step then is the tubular secretion. So tubular secretion is going from the peritubular capillaries to where? From the peritubular capillaries to where? It could be the DCT. It could be the PCT. It could be the nephron loop. So anywhere along the renal tubules. And that's why I named it 2, 2, and 2, because it all occurs along the renal tubules. Okay? So essentially, it's going from being called plasma, and now it's going back to being called filtrate. Okay, so that's secretion. That's tubular secretion. All right, and water conservation is just whenever you bring water back into the body. That's your information. Okay. Now I did this very simplified way because we don't have enough time. But when you're reviewing this, I want you to make sure you also learn about glomerular filtrate rate. Okay. What exactly is being moved? How is it being moved? Okay. And if you get a chance, I highly recommend you watching my class video because I do go through all of that. Okay. All right. So for the male reproductive system, make sure you know the following. Let me get my pen. What are the functions of the male reproductive system? Describe the male reproductive organs, and that includes the testes. Okay, and within the testes, you should be able to identify the seminiferous tubules, the retotestes, and then the cells that form the testes. What are they, by the way? Can you give me a list of the cells that you'd find that form the testes? What are some of the cells that form the tissue or the lining of the testes? Okay. How about germinal epithelial? Oh, there you go. Succenticular cells. Thank you, Amanda. Excellent. So succenticular cells are definitely one of them. And the succenticular cells, they're larger than the other cells, and that's because form they help to block uh, they help to block and protect the germ cells or the sperm cells once they're produced, because once the sperm cells are released, it becomes one end instead of two end, and is recognized by as non-self. So your body would start to attack it. So the succenticular cells kind of provide that protection. Okay, and that's where you'll hear things like the blood testes barrier. Okay, so they're part of the blood testes barrier, BTB. Okay, what's another cell? Interstitial cells, excellent. So the interstitial cells are kind of the cells in between the seminiferous tubules. So what do these cells do? What are these cells responsible for? Yes, they produce testosterone. Okay, and then last type of cell, or you can even tell me the name of the epithelium. Yeah, excellent. So the germinal epithelium, it's what lines the kind of the surface of the seminiferous tubules, and that's where the sperm cells arise or produced. Okay, great. So these are the three main types of cells slash tissues that you would find in the testes. So you have the succenticular cells, the interstitial cells, and the germinal epithelium. Then make sure you know how the seminiferous tubules with these cells extend and become the retatestes. It's just like a continuous tube, starting from the seminiferous tubules to the retatestes, and then those tubes form the epididymis, and then those tubes then become one large tube that form the ductus deferens, passes through the penis, through the urethra. Okay, make sure you understand the spermatogenesis, and that's what we're going to focus on today. And what is the composition of semen and their function? Okay, and make sure you understand the anatomy of the sperm as well. And when I ask you for that, I'm asking for a mature sperm. So one that looks like this. Okay, has three parts. Okay, all right. So we'll start off with the anatomy. So you'll have the testes here, one, and then the C-shaped structure right here that surrounds the testes. We'll number that number two. What is that structure, number two? The epididymis, very good. Then the epididymis extends outward into this tube that goes up. And what is that tube called? The ductus deferens. Perfect. The old name for that, and I'm just telling you so that it makes a little sense when we talk about the procedure, is the vas deferens. So Dr. Vaz is the person who kind of named it, so that's why it used to be called vas deferens. But that is where vasectomies come from, the term vasectomy. So vasectomy is when you tie or you cause a little occlusion along the ductus deferens. Okay, so vasectomy is when you tie off this tube. So the ductus deferens, which used to be called the vas deferens, moves up and around towards the back of the urinary bladder, where it starts to form this area right here. And what is that area called? Yeah, that's the first gland that it will encounter, which is the seminal vesicles. So the tube does move through it, and then it becomes this short little area right here. Sorry, it's a little hard to see. And I'm going to outline it. So it's this tube right here that moves from the seminal vesicles and into the prostate gland. What do we call that tube? Ejaculatory duct, and then it joins up with this tube that connects to the urinary bladder. So what is this tube called? The urethra. Very good. And the urethra is what then moves through the penis, passes through the erectile tissues, and then exits. Okay. Before it does that, though, there's a little bulb right here. Okay. So it does pass through 3 glands. The seminal vesicle, number 4, is one. The prostate gland right here is another. And then this gland right here is the third. What is this gland right here called? The bulbourethral gland. Exactly. And it makes sense, right? Because it looks like a little bulb and it's connected to the urethra, hence the bulbourethral gland. Okay, so relatively easy to remember. Okay, the penis itself is consistent of 2 types of erectile tissue. What kind of erectile tissue would you find that wraps around the penis? So this area right here. So in general, that is the penis, but it is made up of 2 types of erectile tissue. What are they called? Yes. So the corpus spongiosum and the corpus cavernosum. Very good. Okay. So corpus, I didn't give myself enough room. Spongiosum and corpus cavernosum. Okay. And then this last portion right here is referred to as the glands. Very good. Glands of the penis. Okay. So that's the general anatomy of the male reproductive system. Now, before I go into spermatogonium, before I go into spermatogenesis, I want to talk a little bit about reproduction overall. So I'm going to just take a little bit of space right here and talk about Repro 101. Okay, so Repro 101, I have some terms that you should know. Let's start with what are gametes? What are gametes? Yeah, gametes are sex cells. And specifically, you've got the sperm, and the ova. And we use the term egg interchangeably. Okay, ova, egg interchangeably. We have the term chromosomes. What are chromosomes? Yeah, exactly. So chromosomes contain DNA. Okay. So you can see that in a cell, in a human cell, how many chromosomes would you find in a human cell? Yeah. Okay. So there are 46 chromosomes, or what we call 23 pairs, because they're mostly identical. Okay, so you have chromosome 1, chromosome 1, chromosome 2, chromosome 2, chromosome 3, 3, 4, 4, so on. So you have chromosomes, which are strands that contain coiled DNA. Okay, now, so you'll often see something like this. Okay, so here's chromosome 1, chromosome 1. So it's a pair. Okay, so there's one pair, and here's another pair. Okay. And it goes like that from 1 all the way down to 23. Okay. Now, it turns out that chromosome 1 through chromosome 22 are referred to as autosomes. Chromosome 1 through 22 are autosomes, meaning they're identical to each other. So you have two copies of each chromosome. Okay. The 23rd chromosome is known as the sex chromosome. Chromosome. And that's because it could be identical or it could not be. So what are the potential chromosome that you can have for the 23rd pairs of sex chromosomes? Yep. You can either have XX or XY. So it's really the Y that really kind of biologically identifies that it is male. Okay. And because of when you have that Y chromosome, it starts to stimulate testosterone production. Okay. So males can donate either an X or a Y. Females can only donate X and X. So who determines the gender of a child? Yeah. Someone should have told Henry VIII that, right? Before he killed off a lot of his wives. Yep, that's right. So gender is determined by males because only males can contribute that Y. Okay. All right, so let's see if there's any other terms I want you to know. So you know about chromosomes, you know about gender, gametes. I think that's it for the Repro 101. Let's see. Oh, one more thing. Now, with the chromosomes, you oftentimes can see it written as 46 total chromosomes, 23 pairs of chromosomes, or 2N. Okay, so 2N, meaning there's two copies of that one. Okay, however, gametes are different in that they are only what? Gametes are different in that they are only what? Yep, they're only 23 chromosomes total, or else 1N, which makes sense, right? Because when the two cells come together, the sperm and the egg come together, they each give half, donate half from the sperm and half from the egg, which then reunites and becomes a 46 chromosomed cell. Okay. All right. So kind of keep that math in place as we're going through this process. All right. Okay. So now we can start with spermatogenesis. So spermatogenesis, sperm meaning, sperm, genesis, the creation. So spermatogenesis is the process in which sperm is produced. Okay. This process occurs where? Where does this occur? Yep, perfect. So in the testes and more specifically, the germinal epithelium. Perfect. When does it occur? When does this process occur? At puberty. Yep. And so it's going to be from puberty to death. Okay. So a male will start to produce sperm at puberty and continue until they die. Right. And that's why men can have children so late in life, because they're making new sperm throughout their lifetime. Keep that in mind, because when we move on to the female reproductive system, it's actually going to be completely opposite. Okay, so a female is going to be born with all the eggs she will have in her lifetime. And that is actually, that happens when she's still in her mother's womb. Okay, and she doesn't make any more. So that means that as she grows and ages, her egg or her ova also grows and ages with her. And that's why it becomes more chromatic, like chromosome, there's more instability in the chromosomes once a woman reaches a certain age. Usually it used to be 35 and above, but now they're pushing it back to like 40, 45 and above, where it becomes unstable, where the chromosomes become unstable. Okay, but spermatogenesis occurs in the testes, germinal epithelium. It starts at puberty until death. All right, so I just want to take a little step back here so that you can take a look at this right here. So you can see in the testes right here. Okay, if I were to take one of the seminiferous tubules and cut it in half, it would look something like this. Okay. So let me draw the tube first. So here's the tube. I cut it in half, and this is what you're going to see. In the center is the lumen, the space. And then surrounding the lumen, you will have the succenticular cells. And then on the surface, that's where you have the germinal epithelium. So the germinal epithelium, these cells right here, are going to start with the spermatogonium. Okay, and then it's going to divide. One stays in place, and the other one kind of squeezes its way along the in between. Let me use a different color. It's going to squeeze itself in between the succenticular cells until finally it goes into the lumen. Okay. All right, let's go back over here. So here's the germinal epithelium. So in the germinal epithelium, you have the spermatogonium, and that's the kind of the stem cell. Now the spermatogonium undergoes a process called mitosis. So in the human body, we have two forms of cell division. We have mitosis and meiosis. Okay. Tell me, how are they different? How are they the same? Let's go with, well, we know how they're the same, right? Because they're both forms of cell division. Yes. So one divides and one divides so that it can reduce. Okay. So meiosis is a form of chromosome reduction. So meiosis is a form of chromosome reduction, which means then, are your somatic cells going to go under that, or just your gametes? Yes. So meiosis, the point of meiosis is to get gametes. Okay. Mitosis is just a way to reproduce. Okay. So one, you end up with two, and they are identical. So if one starts off with 46 chromosomes, what will the daughter cells have besides being identical? Nope. They're identical to each other, but they're also identical, not polar cells either. They're identical to each other as well as they are identical to the parent cell. So they will also have 46 chromosomes. Okay. So let's take a step. Let's look at that. From one cell that has 46, it becomes two cells that have 46. So what has to happen in this process in order for that math to work? So in mitosis, what has to happen for the math to work from one cell that's 46 to divide into two cells that also have 46? It has to multiply, right? It has to multiply and then divide. So that's mitosis. Okay. So one of your highly mitotic cells in your body is your skin cells. Your skin cells are just going to copy itself and then divide. So the skin cells in your right hand versus the skin cells in your left hand, they are identical genetically. Okay? So mitosis is a reproduction. It's a form of cell division where essentially it's like a copy machine. It makes copies, okay? Identical copies. So if you put one of your original sheet in, you make a copy of it, it's identical to that original, but now you have two. That's what mitosis is. Think of mitosis as a copy machine. You put the original in, you press the button, and voila, you have two. They are identical, okay? Where in meiosis, it's a form of chromosome reduction where it's going to take that cell that has 46 chromosomes, it's not going to multiply it, okay? The first step that it's going to do is it's going to divide. So from 46, and it divides into two cells that are what? Give me a number. If you take 46 and you divide it by 2, what do you get? 23. Perfect. Exactly. That's the first step in meiosis, is that it's just going to go poof, divided in half. It doesn't copy like it happens in mitosis. In meiosis, it just splits it in half. So from one cell that you have 46, now you have two cells that are just 23. And that's where one of your classmates used the term haploids. Okay, haploid is a cell that has half of the chromosome. So in this case, the new cells that are 23 after the first step in meiosis are haploid cells because they only contain half of the original chromosome. Okay, where the cell that has 46 chromosomes, that's referred to as a diploid because it has two copies. Dip, okay, diploid. All right, so that's the first step in meiosis. But keep in mind that meiosis is meant to produce multiple copies of the gametes. So then what's going to happen is that each of these haploids right here are going to produce two more. It goes through this process one more time to produce like this. Okay, now I want you to take a look at that. In meiosis 1, it went from 46 to 23. One cell that's 46 to two cells that is 23. And we said that the only thing that happened there was division. The one cell that was 46 divided into two cells that are 23. Mathematically, check. That works. Now I want you to tell me what happened in order for me to go from one cell that's 23 to two cells that are 23. What has to happen in order to make that mathematically possible? What has to happen in order to make that mathematically possible? That's pretty close. That's pretty close. So instead of using the term mitosis, tell me what's actually happening. Because we know this is meiosis 2, so there's no mitosis, but you're right in that it's very similar. There you go. So in meiosis 2, it's copying what happened in mitosis in that it first copies and then divides. Where in the first step, meiosis 1, it just divides. Okay, and that's pretty much meiosis. Meiosis 1 is a division. Meiosis 2 is a copy and division. Because the point of gametes is to have as many as possible because our biological goal is to reproduce. So in the end, from one spermatogonium, how many sperm cells should I have from that one spermatogonium? Perfect. 4. Exactly. So in meiosis, I start with one cell that's 46, and I end up with 4 cells that are 23. Okay, and now you know how. It starts with a division and then a copy and division. Okay, compared to mitosis, okay, because mitosis purpose isn't to produce gametes. My purpose of mitosis is just to make copies, identical copies. So it's just a multiply divide. Boom, done. Okay, great. I'm so happy. Okay, so now let's apply it to spermatogenesis. Now that you know kind of the mitosis and meiosis. Okay, so in spermatogonium, okay, this cell has 46 chromosomes, or else we can say 2N, or another way we can say it is that it's a diploid. I can use any one of those 3 terms. 46 total chromosomes, 23 pairs, 2N, diploid. Okay, so that cell is completely fine. It's like any other cell in my body. Okay, and then so what's going to happen is that it's going to undergo mitosis. So let me ask you, true or false? So after mitosis, the spermatogonium produces a type B spermatogonium and a type A spermatogonium. Are they identical to each other and to the original cell, or are they different? Identical or different? Yes. Yes, exactly. Because it's mitosis. Okay, it's like taking that spermatogonium, putting it into a photocopier, and making another copy of it. So from 1, now I have 2. Okay? True or false? Mitosis from one cell produces a total of 3 cells. Oh, I love you on it. Exactly. That's exactly it. It's false. Okay? So from one cell right here, it essentially copies the number of chromosomes. So this, just imagine this little, what is this? Here we go. Let me highlight this area. So this intermediate step right here, do you see how I highlighted in yellow? Okay. So in that intermediate, in that time span, the 46 chromosomes has to copy, which means it becomes 92. And then that 92 divides and produces, so it just splits in half to produce 1 here, the type B, and that's why it has 46, and the type A here that has 46. Okay, So in that yellow, in that time span, the chromosome had to copy itself. So it went from 46 to 92 in that same cell, in that original spermatogonium, and then it just splits in half. And that's why from one spermatogonium, the net number of cell is just 2. It's like I took that original piece of paper, I put it into the copier, and I made 1 copy. Okay, so sometimes students think, oh, now there's 3 existing, right? Because here's the spermatogonium, that's 1, the type B is 2, and the type A is 3. No, the spermatogonium, once it undergoes mitosis, it splits in half, right? So that's why you have the type A and type B. So really, from one spermatogonium, the net number of cells I have now after it undergoes mitosis is just 2, not 3. So I don't go like 1 here, 2 here, 3 here. No, no, no. Okay? I go from 1 here, and this one here, and this one here. So from 1 to 1 and 2. So then after mitosis, from 1 cell, I only have 2 cells. I don't have a net total of 3. Okay? Now, the type A stays here, stays within the germinal epithelium so that it can continue and repeat this process, like so, and undergo mitosis. And that's why sperm cells are going to continuously reproduce, because the type A is going to stay there and continue to just like crank out more type Bs throughout time. Okay? Thumbs up, makes sense or does not make sense? Or ask questions.
I just have a question. Is it
Yes,
type A or type B that stays back and
type
continues?
A stays back.
Okay.
Type B differentiates. Yep.
Okay. Thank you.
Yeah. They're identical to each other. So realistically, it could easily have switched, but someone just went, okay, the one that differentiates is now called type B. But if you look at under a microscope, either one of them actually could differentiate because they're identical to each other. So it's not that it divides into a type A or a type B, it's just that once it divides, whichever one goes to differentiate is now called type B. Hopefully that makes sense. Okay, so it's not like it divides and and it goes, okay, I'm automatically type B and I'm automatically type A. Yep. Okay. So whichever one goes on through the process to differentiate and eventually become a sperm, we just indicate that has a type B, but technically they're identical to each other. Okay. All right. So the one that's going to move on is going to begin meiosis 1. Okay. And we just defined meiosis 1, right? So remind me what happens in meiosis 1 again? Perfect. You got it. Yes. A pure and simple division. You got it. I'm so proud because this is hard. Okay. So what you'll find is that the type B differentiates and becomes spermatocyte, the primary spermatocyte. Now, the primary spermatocyte is going to undergo meiosis 1, which is a form of division. So that primary spermatocyte is 46 chromosomes, but after meiosis 1, it becomes secondary spermatocytes. And at this point, how many chromosomes does each cell have? Oh, yes, you are ahead of the game. So each one has 23. You got it. That's perfect. That's beautiful. So 46 divided by 2, the cell splits, just splits. So the half goes to one cell, half goes to the other cell. And that's what produces secondary spermatocytes. Okay. Now the secondary spermatocyte is going to undergo meiosis 2. Someone remind me what happens in meiosis 2. There you go. Okay. So it multiplies first, followed by a division. Okay, so intermediate right here, that 23 chromosome is going to copy and become 46, and then it's going to divide into 23 again. So the secondary spermatocyte undergoes meiosis II to produce 4, get that, 4 spermatids. Okay. So in other words, this first one right here gets chromosome 1, chromosome 1, chromosome 1, chromosome 1, and that's it, just one copy. Chromosome 2, chromosome 2, chromosome 2, chromosome 2. But when we get to the 23rd chromosome, okay, so let's go back to the primary spermatocyte. Of this 46 chromosome, the 23rd pair has to be X and Y, right? And let's just say, for example, when it divides, one of them will carry the X and the other one will carry the Y. Technically, I could switch it around either way, but I know that one will become the X, one will have the Y, because it splits the 23rd chromosome pair apart. Which means the first spermatid that you see on here, what should it be? If this is the chart that I'm using. Okay, let me rephrase that. Okay, so right now, to kind of test, to make sure you understand the process, I'm taking a look at just the 23rd chromosome. Yes, exactly. So this first spermatid would carry the X chromosome as its 23rd copy. The second one would carry which one? Yeah, you got it, X. The third one would carry what? Yep, perfect. And the last one would carry Y. That's exactly it. So the spermatid then will become a mature sperm cell. And you can see the difference, right, between a spermatid and a sperm cell. It's this portion right here. It develops the structure. What is that structure called? The flagella. Excellent. So really, that's the major difference between a spermatid and a sperm cell, is that a mature sperm cell develops that flagella. Okay, so whichever one of these sperms penetrates the egg, that's what determines the gender. So the female only has the X to contribute in its 23rd pair. The male is either going to be an X or a Y that penetrates and determines the gender. Yeah, it's kind of cool, right? That's spermatogenesis. All right. I'm sadly going to go overtime. So if you can hang in there with me, great. If you can't, this will be recorded. Okay. So I'm going to move on to female reproductive. There's a lot more here in terms of like comparing male and female, but I'm going to go backwards and do the female now. And because we're kind of running late on time, I'm going to skip the anatomy. Okay. We're just going to go straight to oogenesis and the reproductive cycle. Okay. So we can go oogenesis and the reproductive cycle. But this is great because it's going to follow what we just talked about in spermatogenesis. Okay. Here we go. Okay. So female reproductive cycle is complicated. All right. And the reason why it's complicated is because, let me put in a table right here. Here we go. Female reproductive cycle is complicated because there are 3 things happening all at once. Oh, I just lost my ability to write on my slide. Give me 1 second. Oh, there we go. So three things are happening at the same time. Same time. That's what makes it so complicated. Okay. Now so the things that happen is oogenesis or oogenesis, and then follicular genesis, and then the uterine cycle. Okay. So oogenesis, follicular genesis, and the uterine cycle. Rather than going through all of them at the same time, okay, I'm just going to focus on one part first, then the next part, then the next part, and then put them all together. Okay, I found that for some reason works better with students to understand this than to go kind of across the different cycles. Okay, we will go across the different cycles, but that's going to be our last step. So our first step is to go through this first, then this, and then pull them all together. Okay. All right. So we're going to start with oogenesis. And it's going to be very similar to what we talked about already in spermatogenesis. Okay. It is, instead of development of the sperm, it is development of the ova. And we use the term egg interchangeably. Okay. Some differences, however, is that oogenesis occurs before a female is born. So in other words, all of this is happening as she is developing. Okay, as the female is developing in her mother's womb. Okay, so I'm going to make this distinction right here. This line. So this line is during fetal development. Or let me see. So this whole process is during fetal development. Okay. So before a female is even ever born, this happens. Okay. She's going to have these cells called oogonias. And the oogonias is 22N, which means it's 46 chromosomes. And it's like, it's similar to the spermatogonia. It's the stem cell. Okay. And this cell is going to undergo mitosis. Remind me what mitosis is again? Yep. It's going to copy and divide. Remember, it's going to produce identical cells. Exactly. Thank you, Savannah. You're way ahead of me. So it's going to undergo mitosis and it's going to produce two cells. One of them is going to stay and repeat this process. The other one becomes the primary oocyte. So the mitosis completely happens when the female is undergoing her own fetal development. Okay, so by the time that she's born, she will have made all the primary oocyte that she will have in her whole lifetime. She will not make any more after she's born. Okay. Now, from the oogonia, okay, one of them will become a primary oocyte, and the other one will go back to being an oogonia and repeat this process until she has enough primary oocyte to last her lifetime. Okay. Now, the primary oocyte starts meiosis 1, but does not complete it. Okay, so meiosis 1 right here begins but does not complete. So technically, when she's born, she still has primary oocytes that have not completed meiosis 1 yet. Okay, so that's why shortly before birth, this whole process pauses. Okay, then it stays that way until what happens? Puberty, excellent. Until puberty. Now with puberty, she's gonna produce a lot of hormones, specifically FSH, follicle stimulating hormone. So at puberty, she's gonna produce the FSH, and the FSH is gonna drive the completion of meiosis 1. Okay, so after birth and then at puberty, she's gonna continue this process and finish meiosis 1. Someone remind me what meiosis 1 what happens in meiosis 1 again? Divide, just divide, right? So it's a way to reduce the number of chromosome. And that's why after meiosis 1, she goes from a primary oocyte that has 46 total chromosomes to N, and it divides into 2 cells that has 1 N each. Okay, so now she ends up with 2 cells, and they're technically called a secondary oocyte. Okay, so the secondary oocyte after meiosis 1 has just 23 chromosomes. But here's a second difference. The first difference is that this process of oogenesis occurs during her fetal development rather than at puberty. The second difference is that in females, it's about quality over quantity. Okay, because where that egg not only contains the chromosomes, but will be the body, the physical body of that first cell that develops once the sperm deposits the male chromosomes. So that means that whatever cell that forms has to be the best. Okay, so that's why only one remains as a secondary oocyte, where the other one, and the cell size here isn't quite correct. When it divides after meiosis 1, one of them is really big, the other one is really small, as just a way to get rid of the extra chromosome. So that's why it's called the polar body. So after meiosis 1, you have a secondary oocyte that's nice, plump, and has all of the the organelles, the cytoplasm, and the chromosome, where the other one is what's called the polar body. Because the polar body is just a way of just getting rid of the extra chromosomes. Okay, so that one dies. All right. So after meiosis 1, you do produce 2 secondary oocytes technically, but one of them is a polar body. In other words, this is a way to get rid of that chromosome, where there's only one that remains as a true secondary oocyte. Okay? Questions? All right. So now this secondary oocyte undergoes ovulation. Okay? So what's ovulated from the ovary is the secondary oocyte. All right? So the secondary oocyte is released from the ovary through a process called ovulation. And this is triggered by the hormone LH, luteinizing hormone. Okay. Now, if nothing happens, in other words, if the sperm does not fertilize the egg, it's the secondary oocyte that a female sheds in menstruation. Okay. So if fertilization does not occur, the secondary oocyte breaks down and is removed during menstruation. Okay. But if fertilization does occur, so essentially, okay. So if fertilization does occur, then that secondary oocyte will undergo meiosis II. So meiosis II never happens unless what else happens? Yeah, exactly. So meiosis II only happens if the secondary oocyte is fertilized by that sperm. Okay? Now, you can just ignore this because the polar body doesn't go through this process. Nothing happens with the polar body, but that's what should have happened. Okay? All right. So, meiosis II, once the secondary oocyte is fertilized, it has to complete meiosis II because that's just a part of nature, right? Cell division, if it's going to undergo meiosis, it has to go meiosis I and II. It has to happen to complete the cycle. Okay. Did it need to happen? Not necessarily because the chromosome has already been reduced, right? But it just happens because that's just normally what happens in meiosis. It's a two-step process. Okay. So when when the sperm fertilizes the egg, the minute that sperm penetrates the wall of that egg, that ovum undergoes meiosis II, which means then it copies and divides. So meiosis II copies and divide. But again, this is not about quantity, it's about quality, right? So then it ends up producing another polar body right here. And that polar body is just a way of getting rid of that extra chromosome that resulted from that copy and divide. Okay, so it goes back to being 23 chromosome. At this point, however, it's going to combine the 23 chromosome from the sperm plus the 23 chromosome from the ovum, and that's what results in 46 chromosome, half from the male and half from the female. Okay, so this is a new cell that results from the chromosomes from the sperm and from the ovum, and that's what we call a zygote. That's the first cell that has 46 chromosomes that is created due to the combination of the chromosomes from the sperm and the chromosomes of the ovum. And then this cell will undergo mitosis until it eventually becomes an embryo, and then the embryo becomes a fetus, and then 9 months later, a baby is born. Okay, questions? So truly, it is an it is an issue of quality and quantity. So in males, it's about quantity. They want their best chance of reproduction is the best chance of continuing their DNA, their chromosome makeup is to make as much sperm as they can. The more sperm they have, the more likely a penetration occurs. For female, for a female to pass on her genetic information, it's about quality. Okay? Questions? So hopefully that helped. That's oogenesis. So we move on to the next one. Is it okay if I move on to the next one now? Or do you have any questions? Okay, I think I have the thumbs up to move on. So now we're going to add the second step, which is follicular genesis. Okay. Now that egg isn't by itself, actually. That egg is surrounded by other cells. Okay, and a follicle is formed around that egg. All right. So we already looked at this side right here. We covered this side right here. Okay. Now I'm going to add on this side right here. And you can see that it's parallel. But it does involve different cells. Okay. So now I'm going to cover follicular genesis. So in follicular genesis, so if this is the egg right here, if that is the oogonia, around the oogonia, I have another set of cells. That surrounds the oogonia. These cells form the follicle. Okay. Now the follicle is also going to change along this entire process. Okay, so let's follow that change, or in other words, the development of that follicle. So surrounding the oogonia, I have what's called the primordial follicle. Surrounding the oogonia, I have the primordial follicle. The primordial follicle, this layer of cells that surround the oogonia, is made up of simple squamous epithelium. Okay. Then you know that during fetal development, that oogonia undergoes mitosis, and as part of mitosis, it becomes the primary oocyte. So the primary oocyte is also surrounded by the primordial follicle. Okay, so regardless of whether I'm looking at the oogonia or the primary oocyte, they are both surrounded by the primordial follicle. So at that stage, that's the only thing that happens. So before birth, in terms of the follicular genesis, I only have the primordial follicle. Okay. It forms around them, and that's it. It doesn't change like it does in the oogonia. Okay. So then at birth, this process stops, nothing happens. And then at puberty, try to remember what stimulates this process to begin again. At puberty, what stimulates this process to begin? Exactly. What does FSH stand for? Thank you. Follicle stimulating hormone. You're probably like, oh, that's what it's stimulating. That's exactly it. It's not that it's stimulating the change in the egg to proceed. It's actually it's changing the follicles that surround that egg. So what stimulates this process every month is technically stimulating the follicles that surround the egg and not the egg itself. Okay, and that's why it's called FSH. Okay, so once FSH is released during puberty, that's when you start to see a change in the follicle, where before that, it's just the primordial follicles. So at puberty, when the FSH hits, that primordial follicle is going to transition and become the primary follicle. Okay, Then the primary follicle is going to become the secondary follicle. And then the secondary follicle will become a mature follicle. Some textbooks have kind of an intermediate right here, which is called the tertiary follicle, where we're just going to go from primary to secondary to mature. Okay, but essentially it's showing you the progression of the follicles. Okay, so here you can see that the primordial follicle becomes the primary follicle. Then the primary follicle becomes the secondary follicle. Then the secondary follicle becomes the tertiary follicle. And then that becomes a mature follicle. Okay. And then ovulation happens. Once ovulation happens, the ovum is kicked out. And then the follicle that remains becomes the corpus luteum. Okay. So that's the transition of that follicle. Primordial follicle to primary to secondary to tertiary to mature, and then finally as the corpus luteum that remains in the ovary after ovulation. Okay, so the best way to do this is to make a table of the changes in the follicle and how do they change. Okay, so for example, for primordial follicle, I would write down simple squamous epithelium. For primary follicle, I would write down simple cuboidal epithelium. Then for secondary follicle, I would write down stratified cuboidal epithelium. And then you can see that between the secondary and the tertiary mature, this water fluid pack forms right here, and that's distinctive of that. And that's called an antrum. Okay, so that's called an antrum. All right, so that's what I would do to kind of help me figure out what changes happen as it changes from the primordial to the primary to the secondary, tertiary, and mature follicle. All right, questions on that? So make a table, kind of list out the changes that will help you understand what does it mean? Why do we call it a primary? Why do we call it a secondary, a tertiary, mature? All right. Does the primary follicle consist of No, it's actually more the primary. It's kind of an odd one, but it goes from simple cuboidal to stratified cuboidal. But it's not fully stratified cuboidal until it gets to secondary. Okay, so what I would write down for primary follicle is simple to stratified cuboidal epithelium. Sometimes, unfortunately, there's not a clear division of what happens exactly when. Okay, all right. Another cool fact is that during puberty, 20 to 25 of the primordial follicle and ovary go through this process. 20 to 25 primordial follicle with the ovum go through this process, but only one is ovulated. So you can see it's a competition it really is about quality. So although 20 to 25 primordial follicle and egg go through this process, only one, whichever one matures the fastest, is the one that's going to actually release the egg in ovulation. Okay. All right. The uterine lining is a little bit more simple. So the uterine lining right here, the uterine cycle essentially has the menstrual phase, the pre-ovulatory phase, the ovulation phase, and the post-ovulatory phase. But essentially, each month, the uterine lining is going to start to prepare in case fertilization happens. It's going to get thick with blood, okay? And it's going to prepare for pregnancy in case fertilization happens each month. And if it doesn't happen, then that layer sheds and begins all over again. Okay. All right. So same thing as the urine formation. I have a very long video on the uterine cycle, as well as the completing the ovarian and the follicular genesis cycle. So make sure you watch that for the full kind of explanation of it. But hopefully this is enough for you to get started and help it make sense. Okay. Oh, question. Okay, what is an antrum? Perfect. Let me go back to that page. So the antrum right here is the space. I kind of highlighted in red right here. This fluid space right here. I will, here, I'll try to highlight in yellow. That's an antrum. So you can see that an antrum forms after the secondary follicle starts to become the tertiary. Yeah, and that's significant because that antrum fills with fluid and it creates a pressure that actually will cause, with the luteinizing hormone, will cause the follicle to rupture and release the egg. And that's what leads to ovulation. Okay. All right. So that is what we have time for today. Do you have any questions any other questions before I move on to the sample question portion? Okay. Thank you so much for hanging in there. Let me open up my quizzes. Let me save this first. Okay, where are my quizzes? Here we go. Do you want to do the male or female quiz first? Let's do the female since we most recently finished that. Anything specific about the endocrine system to look into? So for the endocrine system, I sort of skip that one because it's mostly memorization. So make sure you know the hormones, where they're produced, what do they target, and what are their results. Okay, so those 4 things. Know the list of hormones, where are they produced and secreted, what do they target, and what are their actions. So a nice way to do that is to do a table. Oh, let me show you the table actually. Here we go. Can you see the PowerPoint still? Perfect. Okay, let me go to the endocrine system then. Here we go. So in the PowerPoint that I'll send out to all of you, it will have these sets of slides. So for the endocrine system, make sure you know the function of the endocrine system, know the hormones, be able to describe the role of it. And then here's an example of what I'm expecting you to know. So for each hormone, where is it secreted, what is its target cell, and what is its action? And I gave you some examples of the hormones that are produced in the anterior pituitary gland. So you'll just want to make a table of this and all the other hormones you're required to know. Okay.